sensation of pain coming in and saying, ow, 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 excitatory stimulus, notice this pain. Whereas circuits within your brain that are going, yeah, I already know about the pain and I'm getting a Band-Aid and shut up already. You might have heard of something called endorphins. Those are neurotransmitters used to inhibit the pain. So where the sensory neurons with the pain signal are going excite, the interneurons with the endorphins are going inhibit, and whichever one is stronger wins out against whether the cell has an action potential or not. All right. Anybody want a break before we start looking at membrane potential? We won't get into action potentials and how they work this week, but we'll lay some groundwork. Good to go? Membrane potential refers to electrical differences between two locations. It's potential like potential energy, where if I'm holding something up, they call that potential energy, and if I released it and it falls due to gravity, then it has kinetic energy, which is the energy of movement. So here, this potential energy can turn into electrical energy and send a message. The locations where you have a difference in charge are inside the cell versus outside the cell. Here's a bit of a neuron. The fluid outside the cell is positive in charge. Compared to the fluid inside the cell, which is the cytosol. The difference in charge from one to the other is called the transmembrane potential. In most cells, including neurons, that difference is minus 70 millivolts. How we get that difference is different ion concentrations in the cytosol versus the extracellular fluid. To change ion concentration, ions move. There are three ways that ions can move. An open channel is just, there's the door, go ahead. The ions can cross any time. These aren't very common because all they do is let something equalize in concentration. They let it diffuse until it's the same on both sides. If we want to move ions against their concentration gradient, we need active transport. If you remember this from chapter 3, this is where you spend ATP to push ions against their concentration gradients. Active transport sets up this transmembrane potential, also called resting potential, the difference in charge when the neuron is at rest, not sending a message. There are also gated channels, which are a way to let ions cross with their gradients, but only at certain times. So there's a door in the doorway, and the ions can't open it themselves, they can only go through when it opens. Gated channels are required for action potentials. Two major types of gated channels, classified by what causes them to open, are voltage gated and chemically gated. voltage-gated open when there's a certain change in charge. For example, if other ions get moved and the membrane potential changes from -70 to -55, that could open a voltage-gated channel. Chemically gated channels open when a certain chemical binds to them. It's like you need a key to open the door. And that key is usually a neurotransmitter. Sometimes it's a hormone or some other kind of chemical. But when we're looking at the nervous system, it's usually a neurotransmitter. So this shows a closed gated channel and then an open one, if you can see that well enough. The ions can get through. But because it has a gate, the cell can control when the ions can move. Questions on ion movement? This is why we had to learn all that active transport, facilitated diffusion back in chapter 3. At resting potential, the two most important ions are sodium and potassium. They're not the only ones that are different between inside and out, but they're the ones that make the biggest difference in the membrane potential, and they're the ones that are going to be moving when we open gated channels for them. Sodium is primarily outside the cell. There's a little bit inside, but a lot less in concentration. Potassium is primarily inside the cell. How they get there, you might remember from active transport, the sodium-potassium pump. It spends ATP to push 3 sodium out of a cell and bring in 2 potassium. So it's sort of like sending the siblings to their rooms when they've been fighting. But no, you go over there, and you go over there, and I'm working hard to keep you apart, but it's important to do it. Okay. Notice something about this math? 3 out, 2 in. Yeah, you've got 1 less positive charge coming into the cell for each set of ions you move. And that's why the outside is positive compared to the inside. I don't have a video that just tells us about the resting potential. So I'm going to show just the portion of this one that does, and then we'll come back to it next week for the second part of it that talks about the action potential.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in, while other channels allow some flow of potassium ions back out of the cell. The sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins.
That goes to the next part. Questions on resting potential? Resting membrane potential is also called polarization. It's polar because it's positive over there and negative over there. There are three ways that membrane potential can change. It can depolarize, which means the inside becomes more positive than the outside. Or rather, it becomes more positive. It might go to minus 60, to minus 20, or it could even go to positive. During an action potential, it will go to positive. If a cell has depolarized, you bring it back to resting potential. That's called repolarization. And then there's hyperpolarization, which is getting even more negative than resting potential. So here's our resting potential. Depolarization goes this way. Repolarization and hyperpolarization both make the cell more negative, but repolarization brings it back to resting when it has already depolarized. Hyperpolarization makes it even more negative than resting. We're going to see each of these happening in a certain sequence in the action potential. I think we'll leave off. We'll just do this slide, because then the next gets us into, okay, we'll get the trigger zone too. And then after that it gets into action potential, which I want to leave off until we can just focus on it. So these changes happen in a small area of a neuron, just one section of membrane at a time. That change is called a graded potential. A stimulus causes a change in one little area. What makes it graded is like a gradient, a concentration gradient. Let's say sodium comes into the cell here. There's a whole bunch of sodium right there, but it's gradually diffusing out. So there's a little more sodium than normal, a little further out from that, even less of an increase after that. You get far enough away and there's just the normal amount of sodium because it hasn't diffused far enough. That's why it's called graded. A graded potential can be an excitatory or inhibitory stimulus. We saw excitatory makes it more likely to have an action potential. What it's doing chemically is depolarizing it. So something like this would be an excitatory stimulus. An inhibitory stimulus hyperpolarizes the neuron. It makes the inside more negative, and that makes it less likely to have an action potential. We also said there are multiple stimuli coming in at once. All the graded potentials spread out from the site of the stimulus, and then there's one special place where they all add up. How much has the concentration changed in this spot determines whether the cell has an action potential or not. That area is called the trigger zone. The trigger zone is wherever the axon starts. So for bipolar and multipolar neurons, where the axon starts at the cell body, the axon hillock is the trigger zone. So you'll have graded potentials coming in along the dendrites and cell body, and however much ion diffuses to here determines if we start a action potential or not. What it needs to, the amount of change it needs to make to start that action potential sorry, I forgot to mention on the previous slide where it was shown, is called the threshold potential. And that's often shown as a dotted line. And if you depolarize to that point, then you'll have an action potential. On a unipolar neuron, the trigger zone is instead where all the dendrites come together and start the axon. What makes the trigger zone special is it has voltage-gated sodium channels. So these are the essential structure to having an action potential. Axons have them, the dendrites and cell body don't, so action potentials will only travel on an axon. And the threshold stimulus is that change in charge that opens them.